Hello again. In this second horizontal gene transfer video, we're going to learn about conjugation. So the first video covered transformation and transduction, which were free DNA with no direct contact and phage transfer, respectively. This video is going to cover conjugation, which is direct contact between the donor and the recipient cell. Okay. So this direct contact is achieved through a pilus, a short hollow protein straw that will connect the two cells. The super cool thing about this is, I don't know if you guys remember when we talked about twitching motility that was done by the type 4 pilus, and the bacteria would like reach out the pilus and then it would be like retracted into the host cell, right? And so this conjugation or sex pilus works in that same way. And so the donor cell, which is this top one, because it is giving the plasmid, actually generates the conjugation pilus, shoots it out at the recipient, attaches through protein interactions, and then brings the recipient cell in. So that's nifty. So again, conjugation is direct contact between two independent cells. That is why this is called horizontal gene transfer. And this contact is through the sex pilus, also called the conjugative pilus. So this process only works if the donor cell has what is called a conjugative plasmid. The conjugative plasmid is critical to this process because, so right here, I don't know if you guys can see like that little dark line. This is ORI-T, which stands for origin of transfer. In between this line and this line, so all of this piece of the plasmid right here, those are all genes to make the sex pilus and move the plasmid over. So the directions to generate this conjugative pilus that will shoot over, grab another host, grab another recipient cell, and then allow for the plasmid to be moved, the genes that make that happen are on the plasmid. So the donor cell cannot be a donor cell unless it has the conjugative plasmid, which contain the directions for this to happen. The conjugative plasmid must contain two different types of directions. Number one, directions to build the sex pilus, and number two, directions to move the plasmid. The conjugative plasmid can contain all kinds of other stuff as well in this remaining 2/3-ish of the plasmid, but these two things must be present in the conjugative plasmid in order for conjugation to work. In E. coli, which totally should be underlined, this conjugative plasmid is called the F factor. And so consequently, we're going to be fixing to talk about the F factor on all of the subsequent slides. It is important to know that while the cells are connected, it's possible for other non-conjugative plasmids. So non-conjugative plasmids are small circles of DNA that do not contain these two sections of information. These other plasmids can be transferred while the connection is happening, but because they don't contain the information to start conjugation, a cell cannot do conjugation if it only has these, but these can be moved along during conjugation. Okay, so in E. coli, the conjugative plasmid is called the F factor. This is important. I am not sure why we have this slide. Okay, so we're going to learn about two different kinds of conjugation. The first one is called F plus F minus. Okay, so here is the F plasmid. It is this attractive purple shade. And you guys will remember, let me scoot this some down so you can see it, that the F factor or the F plasmid has to have two things. It has to have the genes to form the sex pilus, and it has to have the genes to transfer the plasmid. Okay, the F plasmid or the F factor has both of those. The F plus cell is somewhat obviously the donor cell that is starting with the F plasmid. Again, the F plasmid is the one with the genes to make conjugation happen. So the F plus cell has these genes to start with. The F minus cell does not have these genes and will be the recipient cell. So at the beginning, we have F plus, F minus. Okay? So you guys remember from the slide before that the conjugative plasmid has a section called the ORI-T. This is where transfer happens. So if you guys were with me, I would ask you, what is the sequence in the DNA at this point? And I would wait a little bit. And then you would say that this is a high AT region, because pretty much everywhere that the DNA is going to be pulled apart is a high AT region. So the ORI-T, the origin of transfer, is a high AT region as well. So the first step, I don't know if you guys, yeah, you can totally see that right here, is a nick is formed at the ORI-T site to make this little tiny gap. And then the plasmid is transported through the conjugation pilus using a super awesome technique called rolling circle replication. So this is it right here. Can you guys see that? Maybe I should zoom in because I'm super excited about this. All right. So here the nick forms at the ORI-T site, and then basically the plasmid turns, and then as the DNA is being replicated, one single-stranded DNA piece gets shuttled through the pilus into the other cell. When that single-stranded piece enters the other cell, then the recipient cell begins to build the complementary strand. So this is the awesomest thing ever. It's very high-tech, and it's in bacteria, of all things. So this is called rolling circle replication because the F plasmid in the donor cell will move around, and then the single-stranded product will kind of go off to the side like that. I hope that you guys think that this is as nifty as I think it is. Well, whether you do or not, it's still nifty. Okay, so that's what happens. At the end, both cells have a successful, complete conjugative plasmid, or in this case called an F plasmid or an F factor. And so to finish, both cells are F plus. Ta-da! Okay, because they both have the F factor, also called the F plasmid. All right, this one is very straightforward. It's pretty easy to understand. The next one is very unfortunately slightly more tricky. All right. So it's so tricky that I'm only going to show you that part to start with. I'm going to actually turn this over. Ta-da! All right. So this uses, ooh, I don't have a brown. I'll use this kind of pink color. All right. So in this case, the donor cell is going to be something called an HFR cell. And HFR stands for high frequency of recombination. An HFR cell has a chromosome, but it has also acquired, I'm going to draw it squiggly, but it's not really, a fractional, can you guys see if I write it here? A fractional F plasmid. Okay. So inside the chromosome. So HFR cells have a piece of the F plasmid in the chromosome. Right? Guess which piece of the F plasmid they have got. If you guessed the piece that has the directions to make the conjugative pilus and to transfer the plasmid, You are totally correct. That is what actually makes this be a high frequency of recombination. And so here we've got, oh, Snuffleupagus. This is totally wrong. All right. This is not a fractional F plasmid. This is a complete F plasmid. That's why it has that. Okay, so the HFR cell has a entire F plasmid in the chromosome. Okay, now we're squared away. Sorry about that. Okay, so the HFR cell has an entire F plasmid in the chromosome. The F plasmid, again, has these genes. So here is what happens. There is a nick forms. Where does a nick form? Exactly. The nick forms at the ORI-T site, the high AT region that is the origin of transfer. So then the chromosome, because that's where the F plasmid is, starts to turn, and a part of the F factor and a piece of the chromosome is going through the conjugation pilus. Whoop, whoop. Here's the problem. The conjugation pilus is not that stable, and the chromosome is comparatively giant. And so before the whole chromosome can go, the conjugative pilus breaks. And so the recipient cell gets a fractional F plasmid and some little piece of chromosome, but does not get the entire F plasmid. Okay. So at the beginning, we have an HFR cell, and we have an F- recipient. No F plasmid at all. Very mysteriously, at the end, we have an HFR cell and an F- recipient because the entire F plasmid did not get moved over, right? And so I've drawn on this one, so I don't know if you can see it. So I'm going to scooch back up and zoom in a little bit. Well, actually, if I zoom in enough, I bet you can see that. Maybe. All right. So there is about this much F plasmid that does not get moved over. So that contains some of the genes to do the thing that makes it conjugation. So to form the conjugative pilus and to move the plasmid. So without the entire F factor, the poor recipient cell remains F- because it still does not contain an entire conjugative plasmid, and thus is unable to start conjugation on its own in the future. So at the end you have HFR, which has chromosome with complete F plasmid, and you have an F- cell, which has chromosome with some, but importantly, not all. I know that looks like mott. Just move on. Chromosome with some, but not all of the F plasmid. Okay. So that is conjugation. Oh, crisis. I need to zoom out some. Okay, this is what happens when I get so excited about the video that I don't always look at the screen because I have to turn to look at the screen. In any case, so there you have it. That's conjugation.